Good morning. I'd like to thank Amy for that incredibly generous introduction and to let you know that I do think of it as a really great honor to have a chance to speak here with you all, as well as the people that are watching online because of our pandemic era. I am going to talk to you today about genome editing, but I'd like to broaden that discussion perhaps to the way in which biotechnology has affected the kinds of things we worry about and the kinds of things we dream about in the 21st century. You know, it was just a few months ago that the Nobel Prize in Chemistry was awarded to these two women, Emmanuelle Charpentier from France and Jennifer Doudna here in the United States at the University of California, Berkeley, for their stunning work on genome editing. Now, genome editing is not completely new. This took many, many scientists and many, many eras of discovery. But basically, it's about trying to make changes in genetic sequences. And the reason why Doudna and Charpentier were honored the way they were is that the use of so-called CRISPR-Cas9, and we can talk later about the biology if you like, was a way to make genetic engineering much more efficient, much more accurate, much more powerful, and very importantly, in many ways, much more accessible, so that you didn't need the same scale of laboratory equipment and expertise to do this work, which meant that not only could senior scientists now do genome editing for one reason or another, but as of today, a mere 5 years after the first major meeting that Jennifer had, we're seeing high school students doing it in high school laboratories. That's how fast this technology has diffused. And when you think about the range of things that you can use it for, that is really some cause for both concern and celebration. You know, genome editing uses, they range from being able to improve on this growing field of personalized medicine so that we can make changes to make sure a medicine works for you in your particular, let's say, kind of cancer. It'll be used in the environmental field in order to adapt plants and animals to the changing climate. There'll be new kinds of industrial products. But the use that has attracted the most attention has been editing human beings. Now, there's multiple kinds of editing. We'll talk about them differently. One is so-called somatic. I can change something in your body today. It doesn't affect your children. It only affects you. Or we can do heritable editing, which means we change sperm or egg or embryo. And when that resulting child is born, the child now has incorporated the change, but it's done so early that that child's eggs or sperm, male or female, will also have the change, and so that genetic change will pass through generations. Two very different kinds of situations, one lasting only your lifetime, another one reverberating down the generations. But to put this in some kind of context, I'd like to step back a little bit. I was delighted when my driver yesterday from the Buffalo airport, and I apologize, I don't know his last name, but Mr. Jeff told me something I didn't know, which is that Thomas Edison had a relationship with Chautauqua and had come here many, many times and had actually fallen in love with somebody here. Well, you know, that fit perfectly with the fact that, from my perspective, looking in a very kind of simplistic way, for the United States, the 19th century was a time to celebrate the inventive power of science. It was electricity, and it was railroads, and it was steam-powered boats, and it was all telegraph machines. It was all kinds of innovations that changed life. Yes, they had downsides, and those of you who are students of history know about them. But in general, there was this notion that science is an inventive power. That began to change in the 20th century. We began to see and began to fear the destructive power of science. We had airplanes that could drop bombs. We had chemical warfare in World War I. And then, of course, probably highlighting everything in the mid-20th century was the atomic bomb. This is Dr. Oppenheimer, who was famous for having worked on developing the bomb with the Alamo Project. And he said, "Now I am become death, the destroyer of worlds," as he watched the atomic bomb test. And that continued even into the 60s and 70s. We began to worry about environmental destruction due to the use of science to create pesticides and other kinds of substances. I know that Elizabeth Colbert was speaking just yesterday about some of the sequelae to those inventions, which were unintended. And I love the fact that the Cuyahoga River actually went on fire seems to me still to be kind of the quintessential environmental moment of the 20th century. But as we moved further into that century, we moved really from physics and chemistry into biology. And the age of biotechnology, for me, kind of starts with Asilomar in 1975. And this is where I begin to think our fear of the destructive power of science morphs into our fear of the creative power of science. This was the beginning of recombinant DNA biology research. It's the beginning of being able to alter organisms. And because there was real concern about the potential ecological effects of releasing, let's say, an altered E. coli, there was a meeting, very famous now, in Asilomar in California. It's a very kind of campy atmosphere, not nearly as nice as this one. And it was almost all scientists with a couple of journalists and one lawyer, I think. And they came up with a kind of charter for their own work in which they said, let's have a great deal of caution and let's make sure that we take extra care to make sure that this work does not get out of our laboratories so that we know what we're dealing with. But they did start doing the work. And their approach to do the work but with careful controls while you watch the work develop, was then adopted subsequently by our national governmental authorities, who then developed regulations about various levels of safety. In the beginning, the simplest recombinant DNA work, again, stuff that's now done in junior high schools, was done in laboratories that were at level 4, where you'd have negative air pressure so that nothing could get out of the laboratory. The air pressure was done so that only things could get in. People wore crazy suits. In fact, in 1987, you can take a look, if you can see, this is a, I don't have a pointer, I'm sorry, but this is somebody wearing what looks like an astronaut suit while spraying a field with the so-called ice minus trait. All this did was protect strawberries from frostbite. That's all. But that image of having to wear a hazmat suit while spraying the field with something incredibly innocuous as genetic changes go is probably what helped to lead to things like the fear of genetically engineered food that we now see with many, many references to frankenfood. And I have to say, the people who are uncomfortable with genetically engineered food are much better at coming up with names like frankenfood, which communicates a world of things just in the name itself, about unnaturalness, about fear, and about the excessive hubris of humanity in making changes in nature. All of that tied up in one word. None of those of us who are comfortable with genetically engineered foods have come up with anything even close to that in terms of effectiveness. The 1980s also was the era in which we began to get very interested in reproductive technologies. We had artificial insemination by sperm donor with sperm donor catalogs. I think I'm the only person ever sent on a congressionally funded tour of California sperm banks. I hope my brother is watching and he's going to cringe right now. One of my brothers had me at a party one time around that era, and I was talking to some of his friends and he pulled me in the kitchen. He said, "Every time I walk by, you're talking about sperm." I really hope you're out there somewhere, Arthur. I brought back catalogs of the sperm donors to the people in my office. I was working for Congress at the time, to see what would people choose. And then we had surrogate mothers and the concern about hiring out women to make the babies that you've designed with your sperm donors. And then in vitro fertilization, where now the embryo itself is outside the body. And that led to lots of opportunities to manipulate embryos. And we began to hear a lot of concern about humans taking control to the point of somehow guiding their own human reproduction in such a way. As if, by the way, when we choose our marital partners and sexually reproduce, we have no control. But that aside, this really was the beginning of moving from the environmental and food-related genetic engineering issues to the reproductive ones. It continues in the 1990s. You probably remember Dolly the sheep, the cloned sheep. She was just a gift to newspapers and magazines around the world. Every one of them came up with one really clever title after another. I was really disappointed with Time magazine. Will there ever be another U? Y-O-U should have been an E-W-U, E-W-E. And even cloning just for basic science research, not to try to produce a baby, which was manifestly unsafe, especially after there'd only been one successful experiment with a sheep, led to a lot of calls to prohibit all cloning of any kind. And there are several states in the US that passed legislation like that. And it was tied up also with some of the controversy around stem cell research, some of which involved getting stem cells from embryos. I used to do a talk called From Stem Cells to Jail Cells to talk about the way in which we have legislated on these topics. So we've got a long history of moving from a celebration of invention to a fear of destruction to now a kind of fear of creation, and a notion that perhaps science is getting out ahead of us, or maybe it's going into areas that it shouldn't go into. And at the same time, it's offering these tremendous opportunities to cure diseases and to adapt the planet to what is obviously a changing situation. And that's why I wanted to then get back to genome editing. It's such a powerful tool, and it makes it faster and cheaper and easier, and fundamentally, in a sense, is democratizing science. Now, let's talk about some of its many, many uses. Some may be frivolous and others serious. Whimsy and art. Now, these are not examples of genome editing. They're examples of older forms of genetic engineering. But what you have here on the left, believe it or not, is glow-in-the-dark sushi. And you can go on the web and you can look for Glowfish Sushi. It's an engineered fish that has a glow, and this is actually a very standard scientific tool to add a gene that will fluoresce so that you can then tag it with another gene you're trying to study, and wherever you see fluorescence, you know that your gene has incorporated, right? So it's a very standard laboratory tool. Here, some people used it for the glowfish that were made for recreational purposes, prettier aquaria, and now made glowfish sushi. It's disturbing to watch them grind up the fish, but it's fascinating. And because they weren't trying to sell it, believe it or not, there's really like no regulatory control on it. Now that said, there's no reason to think it's dangerous. There's nothing in the glowfish that makes it dangerous or more dangerous than any other fish. But nonetheless, I found it surprising. I was at the Food and Drug Administration at the time, and I spent about a day and a half struggling to figure out how to regulate it, and I couldn't because they weren't selling it. And so I couldn't regulate it because it wasn't marketed. Next to it, the glowing nose on the pig. And there was another geneticist in Europe that had a glow-in-the-dark rabbit. Is there something fundamentally wrong about using genome editing to do that? Is it disrespectful to animals to distort them this way? Of course, there are many people that distort their cats and dogs for the annual shows and create breeds that are designed for aesthetic purposes, not for the general, pleasure and joy of the animal itself. So over and over, I find myself looking at technologies and asking, are we really upset about the technology, about the underlying thing it accomplishes, or simply about the fact that it's now easier to do it and maybe more people will try? Is there something that changes when what used to be a very rare occasion suddenly becomes something that lots of people do, with the potential for a wider societal effect? De-extinction and rewilding. Many of you may have heard about George Church. He's a very flamboyant and extraordinarily creative scientist at Harvard who has been working on a variety of things related to genome editing and genetic engineering. And he's been advocating for a long time for us to bring back the extinct woolly mammoth. Now, he explains that there's actually an ecological reason to do this. It has to do with the role that the woolly mammoth played in the far north and in preserving the permafrost, believe it or not. But I think also it was really driven just by this magical idea of bringing back extinct species. There's something really very romantic about that. There's also a tremendous number of ecological concerns about that, since they're no longer in our ecosystem, and it can be challenging to model how they would influence the existing ecosystem for good, bad, or indifferent. Maybe something a little bit less startling would be rewilding. That is, taking things that have been changed in the past through older biotechnology, like hybridization, and to return them to their earlier forms. Many people now go shop at the farmer's market for so-called heirloom tomatoes because the ones that are shipped across the country have been bred through generations of hybridization and crossbreeding to be good for travel. They transport well, they don't bruise, they don't get overly ripe too soon, but they don't necessarily have the same level of flavor that the heirloom tomatoes had, which of course you could only eat when you were local to them and they were in season, right? So what if we could use genome editing to recreate these heirloom varieties, but now we could do it at any time of year? And we can do it in any place. If it was exactly the same, if every gene, if every base pair down to the last one was identical between the one that we created and the one that you picked off the vine originally, they're literally identical in every possible chemical way, would it bother you that one of them had been engineered? I really think this is an important question to ask because it's about whether the concern is in the product. The tomato seems strangely different, but it's not different, so is the concern the process? Is it that you don't trust the process? And we're going to see more and more of that, by the way, in the area of food. Oh, I'm sorry. I thought that I had 2 slides on you, so yeah, there's my great mammoth and rewilding slide. Sorry. Okay. Disease treatment and prevention. Now we get to the stuff that really matters in a very concrete way for all of us here. Right now, there's an experiment going on in Florida in which mosquitoes have been engineered, again, not through genome editing, but that will be the next way to do it, in order to make it impossible for them to pass on dengue fever, which is a real problem in parts of these kind of semi-tropical environments. And there are other kinds of things, malarias and Zika, that would be eligible for this kind of treatment. So you create a species of mosquito that can't transmit it. This has phenomenal potential. And yet, again, we see demonstrations against it by people who'd prefer to have pesticides sprayed on top of them from airplanes to kill the mosquito population rather than release some genetically engineered mosquitoes, even if the technology is one that allows us to stop the spread of the engineered mosquitoes as soon as they've accomplished their goal, something that the so-called Safe Genes Project at the Defense Department has been working on. And then, most directly, disease treatment. And here, the somatic versus heritable becomes important. So, what we'd like to do is be able to identify somebody who's got a disease that's caused by a genetic variant that doesn't function well in their body, and to somehow change that variant to something that's more typical of a person who's healthy. We're already seeing this happen. So here's the first person treated for sickle cell disease. They take cells from her body from bone marrow transplants. Not easy, not fun, not cheap. Outside the body, ex vivo, they edit those cells and they put them back in her body, where they now are generating cells that have the correct version of this gene that controls for the shape of hemoglobin, and suddenly she no longer is suffering from the sickle cell disease and the incredible pain and inflammation and sickle cell crises that are associated with the disease. We're also seeing a lot of interest and some successes in clinical trials at correcting genetically created forms of blindness, retinopathies. So already in clinical trials, we're seeing the ability to take what is a malfunctioning gene and substitute something else that works better. It's almost like when your word processor spell check substitutes a misspelled word for a correctly spelled word. Does that mean it's without risks? No. Putting aside the risk, let's say, of the bone marrow transplant for our sickle cell patient, there's the risk that your edit's going to go someplace that's not correct. We've all had that experience with spell check where it did a great job, except it got to a word that you wanted and mistook it for a mistake and corrected that word as well. That would be an off-target effect, where the editing doesn't get targeted to the place you wanted it to go. Then there's the question of how is the new version of the gene going to operate? Because genes don't operate in isolation. They're embedded in the rest of the genes in your body, as well as all the kind of external influences on how those genes operate. And so we need to be able to predict in your body, when we put in this healthy version of the gene, is it going to coordinate properly with everything else that you have? So there are still risks associated with this, and they're significant. And they're also a problem of expense. This kind of ex vivo editing that was used for sickle cell is incredibly expensive. It requires sophisticated laboratories and personnel. And if you think about it, sickle cell disease, which has its highest prevalence in the US within the population of people descended from African slaves that came here, you're going to find that this population, maybe if we decide to devote a fair amount of public money to it, will be able to benefit. But sickle cell's real problem, its real frequency, is in West Africa, where it's truly endemic. And in many of those countries, they're severely under-resourced. They don't have enough doctors, clinics, facilities, equipment, and expertise to do this. So this kind of ex vivo editing, which seems like it may offer a lot of hope for American citizens, doesn't necessarily provide a satisfactory solution for people in under-resourced places. And indeed, in general, we would like something that's a little bit simpler. So what we're looking for is so-called in vivo editing, where instead of taking cells out of the body and treating them, you simply squirt something into the body and it fixes it, right? Like Dr. Crusher on Star Trek Next Generation. And, and even Dr. McCoy in the earlier version was able to just press, I don't know if you know this, it was actually a salt shaker that they used. That's what the set dressers used. He would press a salt shaker against your arm and suddenly you'd be cured. So in vivo editing in many ways is the goal. Now that's not easy. There are body systems that are more amenable to this. Things like blood-related diseases you might be able to attack a little bit more easily, and even the retinopathies. But other diseases, it may be very difficult to put something in the body and make sure it goes to the right place and only to the right place. So we're far away from it. But there was a major advance just in the last couple of weeks in which, for the very first time, we saw a success for in vivo editing, again, against a blood-related disease, this transthyretin amyloidosis. Now, it's just the beginning. But if you think about this, not only for yourselves as you get older, but for your children and grandchildren, we are really looking at a true revolution in how we think about treating the body when it comes to things that are genetically related. So this has vast potential and is really what is exciting the field so much. Despite that, what's really upsetting the public more than anything else is the idea of heritable germline editing of embryos, or sperm and egg in a different version. This is a picture of Hu Zhongqi. He's a Chinese scientist who decided he was going to try to edit embryos. And he said he was doing it for a good purpose. He wanted to give these children an enhanced resistance to HIV, which causes AIDS. It was foolish for, I can't even count how many reasons. It was foolish because what he was doing wasn't necessary. These children were not going to be born to mothers who were HIV positive, so they weren't being exposed to HIV in the womb. And if you're born healthy and then later develop HIV, we now have good drug regimens to help you out. You don't need to do something crazy and scary and risky like this. The second is that we had absolutely nothing near to the level of basic science research that you need to even think you have a good way to predict how well this is going to work and what level of risk you're facing. There's many people who would oppose this kind of heritable editing for other reasons, and I'll mention them in a moment, but in this case, just safety and effectiveness alone would tell you that this was absolutely an irresponsible kind of experiment. But he really brought home to the public that this technology is real, and people began to imagine that suddenly everybody's going to want to use genome editing to design their babies for all kinds of things, frivolous and serious, or even impossible. I mean, people began wondering, are we going to edit our children for intelligence, as if we know what makes up intelligence, let alone how it's controlled genetically and through other influences. But nonetheless, magazines began to run all kinds of articles about the fear of designer babies. In some ways, even more than the concern that this would be unsafe for the children, or that it was taking away their choice, or that it treats children as commodities instead of gifts. I think that it really touched on something deeper. If a scientist is doing this, which is a slide from in vitro fertilization, is it conveying a message about this? I really got the feeling that at some very deep level, what's happening is that there is a concern that somehow we are now in the world of science doing things with regard to creation that are really not supposed to be within the realm of human control. And that this really then becomes the source of the anxiety that leads to all the more kind of pragmatic concerns about risks and benefits and consent. That it's really about this idea that somehow we shouldn't be playing God. That's the phrase you hear all the time. And I do want to just take a moment here to mention that that phrase playing God in itself is interesting because it does touch on a particular kind of religious or spiritual view in which a deity actively controls events so that when humans control them, somehow we are now taking on that role. There are other religions in which the deity does not have that kind of active control, religions where there's no petitionary prayer, please do something or other, because there's no notion that the deity has that kind of control over events. Deity creates the world, and then after that, it just rolls along. In Mendel's Dwarf, which is a marvelous, although I have to warn you, very vulgar book, the protagonist is a hereditary dwarf geneticist. And he's looking at embryos in a dish, and he's looking at them, realizing he has the power to decide which ones to bring to transfer for gestation and bring to birth. And he's deciding whether to pick the ones that would look like him and be dwarfs, pick the ones that don't have the dwarfism trait and would have average height, or just randomly, pick all of them and then let statistics choose. And he has this revelation that for him, the deity actually is the one who just rolls the dice. The deity is the one that sets up a system in which 1 out of 4 of your children will have this particular condition because you and your partner both had a particular recessive trait. He says, if I choose, it's not being God, it's being human. A complete reversal of the way that phrase playing God is understood in most of the ways it's used. And I point that out not to say that one view or the other is right, or that one religion or the other is right, but mostly so that people can look at that and ask themselves, how is somebody using that phrase? We're about to see many more developments that are going to challenge this notion of creation and the rightful role of human beings. This is a slide about so-called sheafs. The idea being that you could take cells that would grow in a way that resembles human embryos enough that you could do research on them to understand basic cell biology, differentiation, but that they wouldn't be real embryos in the sense that they could never, under any circumstances, develop into children. So here is the dilemma. The closer you get to being a quote-unquote real embryo, the better you have as a model for your research, but also the closer you get to the idea of creating embryos just to do research on them, which tends to be the one thing that even people comfortable with embryo research find themselves stumbling on. So that's about to happen, or actually it is already happening as embryo models are being developed, and indeed the International Society for Stem Cell Research, and just in terms of revealing biases, I was on their steering committee for this, wrote a new set of guidelines that included some attention to this question about what level of model is appropriate and what can be done with them. And truly, it was the viability of that embryo model to actually be an embryo that could develop that seemed to be the significant kind of dividing line, but knowing where that dividing line lies becomes more complicated. Then you have the ethics of experimenting with brain tissue. I noticed that you're going to be, for those of you who are here, you're going to be hearing from Nita Farahani, one of my bioethics colleagues from Duke University, in a couple of weeks. She's a real expert on the ethics of neuroscience. But we are really beginning to look more and more at the ability to grow so-called neural organoids. that is to grow collections of cells that will mimic some brain functions. It's widely misunderstood to mean growing brains in a laboratory dish. We're not even close to that. But it did make us have to ask a lot of questions about the nature of consciousness. What's the level of development at which you do need to start worrying? We're not there yet by any stretch, but when do we worry? What's the collection of neurons that are needed? How do they have to interact before you can have anything that constitutes some philosopher's notion of consciousness? And even more fundamentally, what does it mean to be human? You know, we're learning more and more about the amount of our genetic heritage that comes from what we used to think of as different species or subspecies like Neanderthals and Denisovans. And now we're already, and this is just from I think yesterday or the day before, we're already seeing articles talking about how we may have to redefine the tree in which we kind of give names to Homo sapiens. We may be Homo sapiens sapiens, but some of these others may also be Homo sapiens, just a slightly different version, and not off on the tree completely. So we're really struggling now with what it is that makes us human, and the more that genetic engineering and genome editing could change a couple of things here and there. Can we switch ourselves from one subspecies to another? I mean, it makes you ask, why do we care? What makes it important, and why is it fundamental to our notion of ourselves and our identities? I want to get close to my conclusion here with something that's going to be particularly disturbing to some people, or to many people. You know, we already have people working on creating chromosomes, just synthesizing them out of chemical materials, so-called artificial chromosomes. And they're very, very useful for genome editing when you want to put a large piece of DNA, right? That'll, let's say something creates a whole cascade of enzymes. So you synthesize this long stretch. Okay, that's great. But you know how DNA is made up of the bases, and we're all familiar with those bases? Yeah, then there are people that created DNA with new kinds of bases that have never been seen before in nature here on Earth. So now we're creating a new genetic alphabet that might represent how life develops on other planets. And there is a whole field of astrobiology that is incredibly interesting, and I know you're going to hear again tomorrow, I think, from somebody who works on space exploration. It says something about whether the four particular chemicals that evolved on Earth are special, if there's something significant about them, if they're unique. Maybe not. Maybe we're not that unique. Maybe we're just an accident of 4 chemicals as opposed to 3 or 6 or 10, and another planet would have a different combination. There's one author who asked a very provocative question. He asked, if we find life on other planets made in different ways, what does that do to our religious beliefs about our own origins? And in a way that I apologize, this is not me, this is him, but I want to just let you know what he's saying. He uses Klingons from Star Trek. If Klingons are living on a planet, they couldn't suffer, another planet, they couldn't suffer from original sin because they're not descended from Adam and Eve. Christianity would make no sense for these creatures unless our understanding of original sin makes no sense. Now, agree or don't agree, but he's asking a very interesting question about the creative power of science now here on Earth to create organisms that we have never seen before through synthetic biology. Combinations that we've never seen in nature, combinations nature could never make, or using chemicals that don't even exist or have never been used. And so, at that point, what does that say about the way we think of our own origins and creation? Does it challenge those really cherished ideas and beliefs? Because in a sense, with synthetic biology, you can say, now I am become life, creator of worlds, not now I am become death, as Oppenheimer had said. Craig Venter is one of the more provocative scientists who's been working on trying to actually create life completely artificially. He's not gotten there yet. He still needed to use certain existing organics, like cells, that he could then populate with artificial structures. But he's working toward it, as are others. So he's looking to try to see, can you make life out of nothingness? And what's particularly interesting is that in the articles describing his work, they describe it as a program that's driven by explicit atheism, an attempt to prove that you do not need a deity in order to create life. That, to me, is something that really touches very deeply in people's hearts and beliefs, but is a question that really is something that we must begin to grapple with now that science has powers it's never had before. Now, it's not that we've never before seen the idea that some research could actually, in a sense, be blasphemous. You know, centuries ago, when there was the debate around whether the sun revolved around the earth or the earth revolved around the sun, we saw efforts to censor these very disruptive ideas about the centrality of humanity in the universe. This is a quote from Goethe, which explains, I think, a lot about what's going on today as well. He wrote, "The world had scarcely become known as round and complete when it was asked to waive the tremendous privilege of being at the center of the universe, and no wonder contemporaries didn't wish to let this go, offered every possible resistance to a doctrine which in its converts authorized and even demanded a freedom of view and greatness of thought so far unknown, in fact, indeed, not even dreamed of." Very challenging ideas, very difficult ideas to grapple with and to integrate into your existing worldview. I'll end only with this, because each of us has to come to some kind of view on this for themselves. For me, I take my direction from Thomas Jefferson, who wrote that liberty is the great parent of science and of virtue, and a nation will be great in both, always, in proportion as it is free. Thank you very much.